Here we go.
Okay.
I guess good afternoon to you. It's morning here in California. Thank you very much for the invitation to come and talk to you today a little bit about what I call an asymmetric career path. And I think since my career has been going on longer than many in the audience have been alive, hopefully I can share some stories, little vignettes from bits of it that, you know, we'll see if you find it useful. This is a picture of where I am now. This is Scripps Research Building, and we do get rain in California. So this is a picture of a rainbow that went from my building, started actually right near my office there, and goes across the parking lot and ends. The pot of gold apparently is at this sculpture, which is called the Flame of Knowledge. So I always find that an inspiring way to start a lecture. So I mentioned my career path is quite long and varied. So I'll just give you a timeline first. I started out in chemical engineering. All my degrees are in chemical engineering, and I was a professor at the University of Pittsburgh and did a couple of sabbaticals during the time I was there, one in France and one at Princeton. And then I made a move that's quite unusual, I think, for academics. At least in those days, it was even more unusual than today. I left and I was recruited to go to Merck Pharma Company at a time at the height of the anti-HIV protease inhibitor development. I'll talk a little bit about that. To start a new group there to really try to understand catalytic reactions in more detail. And from there, I made another big leap, which was back to a more academic group, but across the ocean to Germany. I was at the Max Planck Institute for 3 years. And then I came over to England. I was a professor and chair of physical chemistry at University of Hull for 4 years. And during that time, I did 2 sort of half sabbaticals, one at Harvard and one at Berkeley. From Hull, I stayed in England this time and went to Imperial College London. And there I had a joint appointment in chemistry and chemical engineering and a chair in catalysis. And I'm still a visiting professor there now. But in 2010, I left and came back to the States, to California, which is a little different from the East Coast, but where I grew up. And now I'm the John C. Martin Endowed Chair in Chemistry. And I'm also the head of the Department of Chemistry at Scripps. So I'm just going to give you one small vignette from each of these experiences that I put together to sort of talk about my career, how it's changed, and then maybe to bring up some questions about how you make decisions in career moves. So I started at the University of Pittsburgh, like I said, and I had what was called it's now called a career award from the National Science Foundation. It's a 5-year program for young investigators. It was called Presidential Young Investigator at the time, and I actually have a certificate signed by Ronald Reagan. So, but at that time I was working in I started out in heterogeneous catalysis, and a lot of the work was sponsored by General Motors. Actually, as part of this award, you get matching money from companies, and we worked on trying to understand the chemistry at the surface of palladium, rhodium catalysts. Supported catalysts for catalytic converters. It's a fascinating engineering problem. Basically, you're trying to do oxidation and reduction at the same time. You've got to oxidize the CO and reduce the NOx's. And so these 3-way catalysts they swing between this very narrow window between oxidation and reduction. And so it's fascinating for the engineering aspects of it. That's why fuel injection is so important. And I learned a lot of things. I learned, for instance, don't run out of petrol in your car, because if you do, this catalytic converter will swing way over to the oxidation cycle and you will burn up your catalytic converter, and you'll hear the metal pieces rattling around the next time you drive. So very interesting. But I had always been interested in organic chemistry, and this is a question that is just about reactivity. It's yes or no. Do we oxidize? Do we reduce? And there's no organic molecules here, and not a lot of selectivity issues, which was something I liked. So as I mentioned, I went to France during that time, and I worked at the Institute of Catalysis in Lyon. And there I got into a little bit more organic reactions, although nothing like what my pharmaceutical friends' molecules look like. This is not much bigger molecules. But this is a selective hydrogenation reaction, still with heterogeneous catalysts at this point, but just trying to make the catalyst decide whether to go to hydrogenate a CC bond versus a CO bond. And so you're playing around really with thermodynamics and kinetics. If you want to try to control the reaction, You need to get kinetic control if you don't want the thermodynamic product. So this really, this is the kind of work I really like, because you have to play around with how do I make it go one way or another. And actually this interplay of thermodynamics and kinetics goes all through my work. And the best comment I ever heard is someone trying to explain the difference between thermodynamics and kinetics. And it's a paraphrased quote from the Bible: "The meek shall inherit the earth, but not soon." And so that's why we like kinetics, because we're not meek. So, so this was, I brought these projects back from France, and I was working in further work on these selective hydrogenation reactions at a meeting of Organic Reactions Catalysis Society, when some people from Merck were at the meeting. And they came up to me after the meeting and said, "We have this idea. We'd like to set up a new group that will have a dedicated effort," and we call it the Catalysis and Reaction Engineering Lab, because they realize that pharma companies don't spend a lot of time. They spend a lot of time on drug discovery, on what molecule they have to make. And then the rest of the process is very compressed, and they don't have a lot of time to understand the chemical reactions. And they thought they would do better if they could do this. And it was really brought on by the huge magnitude of the AIDS epidemic and the crisis of making drugs for that. So they asked me to start this group, and I jumped in, had no idea what I was getting into, but it turned out to be a really good move. And many other pharma companies since then have come up with these specialized groups. And I actually helped several companies start groups like this. AstraZeneca, when they were in the UK, as well as Pfizer in the US. And it caught on even with the FDA. The FDA, it took them a little while, more than 10 years after we had been doing this at Merck, but they come up with terms like quality by design, and they actually let you now get your process validated based on understanding, rather than just sort of rote, we did 3 batches and we got the right result, therefore it's okay. It's really based on things like in situ probing of reactions to get process understanding. So, and we worked on, like I mentioned, this was during the time, the early 90s, when HIV was a death sentence if you got HIV. And now it's actually a manageable chronic disease. But back then it wasn't. And they were just starting to develop these protease inhibitors, which the clinical trials were saying were really good. But the problem was they had to make huge amounts of these drugs. Patients had to take 3 grams a day. And pharma companies just weren't used to that kind of production level. Their production is much smaller than, say, chemical companies like the ASF. Someone had done a back of the envelope calculation that said if Merck had stopped making every other drug they made everywhere in the world and just tried to make Crixivan in all those plants, they wouldn't be able to make enough. And so this is what made them a little worried, and actually was the driving force for process understanding, with the idea that if we understand what we're doing, we might actually do it more efficiently, which was a novel idea for pharma at the time. So this was the molecule that Merck was working on. And one of the steps, I just highlighted the part that I worked on here. One of the steps was starting with a pyrazine carboxamide, and you want to get to the piperazine. What you wanted was the S-piperazine. You needed only, this is a chiral molecule. We needed only the S, the left hand. And so 2 things one could do is to try to make it as fast as you can all the way to the end and then separate, or try to stop at C, which is a prochiral intermediate, and then put in a chiral catalyst to get to the S product. And we actually looked at this project both ways, but they had this idea that they had to find a magic catalyst to stop the reaction at C. And if you know, again, about kinetics and thermodynamics, hydrogenations are downhill. And they would basically, the way they started looking at this before we got involved was like 18 Johnson-Matthey catalysts in par shakers overnight, come in the morning, and they had D. And they didn't understand that they needed to think about kinetics to get, if you wanted to stop at C. So this was early on in the days of in situ reaction monitoring. People didn't have React IRs on their bench. We had one in my lab, but it was actually really early on in the days of, Mettler Autochem was not even selling them yet. It was a company called ASI. And so we basically took a look at this reaction, and we were able to see it. This is just under one set of conditions. I think this was 70 degrees and 70 PSI, the starting material goes away, and the product comes in, and then we had one intermediate, this species here, that would come in and then go away. We didn't know which one it was. And the question is it B or is it C? And we hoped that it was C, because if it's B, that would be bad news, because then if there's only one intermediate, B to C to D would be going very fast. So we basically looked at this with IR, we looked at it with reaction calorimetry and hydrogen uptake measurements. And but at the point where we had a maximum amount of our intermediate, whichever one it is, and almost no product, we could calculate the amount of hydrogen that we had taken up per mole of substrate. And we could calculate from the heat flow how many bonds had been broken. And both measures said 2 moles of hydrogen, 2 bonds broken, which told us that we had made it to C. And that was great news, because then we just built a kinetic model to maximize that green line intermediate. And we did all this without actually sampling from the reactor, because the project was so early that even the analytical department was still trying to figure out, HPLC analysis for this. So we did all this just from 3 orthogonal in situ tools. And then we just built a simple kinetic model from the early experiments and played around with the model, saying what conditions we needed to maximize this species C. And you can see this was, I think, was 35 degrees C and ambient pressure. We were able to make almost quantitative yield with almost very little loss to the racemic product. We could capture 45 grams a day in the RC1 500 milliliter reactor. So this really opened the eyes of a lot of the organic chemists about how reaction understanding can be a design variable. They think about designing the catalyst and the ligands. Organic chemists tend to think about that, but a kineticist and an engineer can think about, how do I design the process to get the right result I want? So we started to do, and so again, it wasn't just a magic catalyst. It was really putting the catalyst together with the process understanding and the kinetic model. And we probably could have done that with any number of those Johnson-Matthey palladium on carbon catalysts to get to C. So we started then looking at what to do when we had C, and Merck did develop a process for the asymmetric hydrogenation. Kai Rossen did, it was a phosphane catalyst, I think. And so we started looking at model systems in asymmetric hydrogenation. Now we're talking about homogeneous catalysts, just to try to understand pressure and temperature effects better. And this is a ruthenium binap system that was studied by Naori for many years. And Naori really gets the idea of kinetics. He said, I love this quote from his book. He said, asymmetric catalysis is 4-dimensional chemistry. You have to not just have the structure, lock and key, you actually also have to have kinetics that fit. One of them has to go faster than the other to make a high EE product. And the other quote that I like that sort of tells you about kinetics is from the late Jeremy Knowles, who was a professor at Harvard. He came from Oxford, and he said, you can't just look at the photograph of a racehorse and learn how fast it can run. You actually have to ride on it. And that's what we talk about with in situ tools. We ride on the reaction. And so we can find out how fast it runs firsthand. So with this system, we found something really, really interesting. Most people, when they design asymmetric catalysts, you take the left hand of ruthenium binap to do one thing, and you take the right hand to do the opposite product. Well, we found that we could take the right hand catalyst, and just by changing temperature and pressure, we could make high EE to S, or we could make high EE to R. And all it was that they had a very different profile for whether the system liked low or high temperature and pressure. And one of the first times I gave a talk on this was in Strasbourg. I was already in Germany by the time I was talking about this. And John Osborne, late John Osborne, was the person who invited me. He was the student on Wilkinson's catalyst with Jeff Wilkinson. And he had a long career in asymmetric, or in organometallic catalysis, but they tended to think very much about thermodynamics. At the end of my talk, after showing them this, he stood up and he said, "Thank you for a fascinating and frightening talk," because it's quite frightening to a chemist who's thinking only about, lock and key. How do I fit the substrate into this structure? And telling him that I can make it go either way, with not by changing the catalyst, just by changing the conditions. So that was work. That was pretty much the last work we did at Merck. But we had also found at that time this really interesting side reaction with ruthenium binap. Rhodium binap undergoes isomerization pathways. In fact, that's the main step in the Takasago's process to make menthol. It's a rhodium catalyst, isomerization of geranylamine. We thought, we found this process where geraniol can isomerize to its homoallylic alcohol. We thought maybe this could, be something that could help Takasago in their menthol process. And I met with the head of Takasago's research, came to my lab in Germany, and first thing he said is, "Did you smell the reactor?" I'm like, I don't usually stick my head in the reactor. And he said, no, gamma geraniol is important on its own. It's actually a very important perfume chemical. Different perfumes have these different, you talk about base note, heart note, top note, but they all have some chemicals that they start with. And gamma geraniol is really important. And it turns out the way they make it now, it's with a borane reduction. And you could get it to 99.7% pure. And there's still a slight odor from the boron that's remaining in there. And so I made our lab made some from that borane process, and we made some from this process, and we distilled them. So we had 99.3% pure this way and 99.7. And he came in, and he's head of research at Takasago. He comes in, he pulls out of his pocket, those perfume sticks, like when you go to H&M or something, and he takes some from one vial, takes some from the other vial. He said, this is the gamma geraniol, this is your process. He could tell the difference at 99.3% purity, which was which. So I'm thinking, this is opening up a whole new career for me. I'm going to start sending my grant proposals with scratch and sniff patches on them for people to smell what we're making. But anyway, so that they've other perfume companies also, Firmenich in Switzerland, have been looking into this process because it opens up a lot of possibilities for perfume chemicals. So after that, also at the Max Planck Institute, I started to get into another area of asymmetric homogeneous catalysis, which talks about nonlinear effects. Nonlinear effects are, the EE that you get of the product, the enantiomeric excess. This is a fraction. So 1 is perfect, 0 is racemic, equal amounts of both. And then you look at the catalyst, you compare that to the catalyst EE. Typically, you try to make your catalyst enantiopure. But sometimes the process isn't all that easy, and you might not have an enantiopure catalyst. But there have been a number of, and you expect each catalyst would just do its own thing separately, and you would be on this linear path. Like if you had a 60% EE catalyst, you would expect 60% EE product, or proportional to the catalyst. But there are cases where you get amplification and deletion, and nobody likes depletion. You don't want to get less EE than you paid for with your catalyst, but amplification got everybody excited. It sounds like a free lunch. I've got a 20% EE catalyst, and I'm making nearly the same EE as if I'd spent all that time making 100% EE catalyst. And it turns out it isn't a free lunch. What you pay for is in the time it takes you to make the product. For instance, if you look at how much you would make in one day of the correct enantiomer, in this depletion case, to make the same amount in the amplification day case would take you 15 months. Okay, that's an apocryphal example, but what it shows you is what happens is that higher order species are involved, and this is Kagan's dimer model. When you put in your catalyst, you counted your EE of the catalyst as how many S's I put in, how many R's. I put S's and R's in the reactor, but they make dimers, and they can make homochiral dimers or heterochiral dimers. And the ML2 model in this case is essentially the RS heterochiral dimer is either inactive or much less active than the others. The SS will make one hand, RR will make the other. But if you start with almost equal amounts of, like low EE of the catalyst, then you have almost equal amounts of S's and R's, and you pull off. In order to get high EE of the active catalyst here, you have to pull a lot of it away. So it's an in situ purification of your catalyst, and you end up with almost, like 90% of your catalyst can be sitting here as an inactive RS. So, it looks great when you get a high EE, but that's how you pay for it. But there are cases where paying that price actually makes sense. This was another Merck process that I worked on later in Germany. I knew it was going on when I was there at Merck, but we did the kinetic modeling of this when I was in Germany. And this is a process that's a air alkyl ketone reduction for this asthma drug, and which is now on the market. But essentially, you use a chiral reagent. It's not a catalyst. It only does its job once and then it crashes out. So it's called a chiral auxiliary. But basically dip chloride is what this is called. And it comes from alpha pinene, and it can make, LL, DD, and LD. It makes these dimers. So it's perfect for looking at the Kagan nonlinear effect model. It turns out the dip chloride comes from alpha pinene, a natural product, but you can get it in two purities. You can get it at 97% EE and it's really expensive, or you can get it at 85% EE and it's really cheap. So the question that Merck wanted to ask was, can we use the 85% EE? Can we still get high EE in our reaction product? And so we developed from their experimental data, we developed a Kagan model, and we could look at a bunch of different EEs. This one just is a case of looking at 50% EE. But basically the model tells you that how much you start out with each of these dimers. And it tells you that the thermodynamic way they proportion themselves. And the K value is the equilibrium constant is 49, which means essentially that we have a little bit more DD, almost no LL, and some DL. The DD is the fast reacting one, and it reacts away. The DL can actually react. It's not inactive, but you can see it reacts a lot slower, and it's making racemic product. So our EE is doing pretty well here until the point where our DD runs out. And then most of the rest of the reaction is going to be from making racemic product. So the rate goes down at rate about 60% conversion, and the EE goes down. So the model can tell us how to prevent that. How much extra of the dip chloride do I want to put in so that this line doesn't go to zero until I'm at 100% conversion? So I don't have to use any of the DL, the racemic guy. And so essentially the kinetic model predicted the number, and it was like 1.3 equivalents. And so they're running this process with that, and the price they pay is a little bit extra, 1.3 equivalents instead of 1 equivalent. But if you're buying it at 85% EE, you're saving a lot of money there. And so 1.3 equivalents is worth it to not have to pay for the expensive. So that's often something that happens in pharma research, is you have to play off, how much something's going to cost you. Sometimes the elegant chemistry doesn't always win if it's too costly. So then when I moved to the UK, an area I got into, which I found fascinating, is the origin of biological homochirality. Okay, and this came about because I've been working in nonlinear effects, and that to me says that's the ultimate nonlinear effect. The prebiotic soup had to be racemic, equal left and right molecules. How do we get where we are now, where all our building blocks are in biology are single chirality? And there have been a lot of theoretical work, but not a lot of experimental work. And there was this paper by Frank, who was actually at Bristol, at Oxford and then at Bristol. He was a crystallographer, but he basically said, if you have an autocatalyst, if you can make yourself and prevent your other hand from being made, or just to suppress it, that's a simple and sufficient life model to get to homochirality. And the last sentence of this theoretical paper, he said, an experimental demonstration may not be impossible. And so chemists try, this is 1953, good year for actually origin of life research, but Hans Winberg in Groningen wrote a review of this field because he was working to try to find that experimental demonstration. He said that sentence was a challenge to any red-blooded chemist. And in fact, the challenge was met in 1995. Kenzo Soai developed this reaction called Soai reaction now that basically the reaction product catalyzes its own formation and the EE goes up over time. So I knew that the kinetics was going to be important here because you're changing the amount of catalyst in the system with every turnover. You can't do initial rates and get anywhere, right? So we developed a kinetic model together with John Brown at Oxford. John Brown sent Chris McMillan, a part 2 dissertation student, to my lab, and Frederick Brono worked later and was a postdoc in my lab. And this is just a couple of our early runs. We developed just an ML2 model. We had the Kagan model for catalysis. We had already modified it for the system I just talked about, which was synthesis. Catalysis, the same molecule does the job over and over. Synthesis, that molecule does the job once and it's done. And autocatalysis, the molecule makes more molecules to do that job. So we just made some more modifications to that same model. And that's what's in pink is the model fit to 2 different runs in the Soai reaction. This is just fraction conversion. It doesn't say EE yet. It just says, the model, we asked the model to find the best parameters to fit this. these data. And then from that, the model can predict what the EE should do over as conversion goes up. So this is product EE for the 6% and for the 22%. Based on the model. And then we can take data completely separate from the kinetic profile data. We can take experimental HPLC data, and that's what we got. So we felt like we were on the right track with this model, that we could actually predict the amplification of EE in the SOI reaction solely from the kinetic data with this model. So it was, but one of the things I want to say to some of the younger people here, if you, whether you're in academia or wherever you do your work, sometimes you have to go against the grain. And when people tell you something's not going to work or they don't think it's important, if you believe in it, keep going. So I submitted a proposal to EPSRC, the first one when I got to the UK, and one of the reviewers said this proposal should not be funded because all the PI is attempting to do is to study an already discovered reaction. Okay, so I think this is a synthetic chemist who's like, discovery is everything. Who cares how it works, you know? And so with EPSRC proposals, you're allowed to send a page of rebuttal. And this was my first, I probably wouldn't be this cheeky later, but I don't know why. But anyway, this is what I said. If the apple doesn't fall on my head, I'm not allowed to study gravity. So I got the proposal funded. EPSRC said, you know, the PI provided a robust response to that comment. So stick to your guns if you think you have a good idea. So, moving on to, well, actually this slide really goes through everything from Merck, actually from my training as an engineer on. We developed reaction progress kinetic analysis, and we had a couple of review articles published. Actually another one in JAX more recently. But basically using these in situ tools to extract information much more rapidly, streamlining process research and also academic research. And Mettler has now developed what's called isokinetics, so that the software can do all these plots that we developed in reaction progress kinetic analysis. And we worked with a whole bunch of people. It was just, I mean, I've had some amazingly wonderful collaborations. Eric Jacobson at Harvard, Steve Buchwald at MIT, my colleague Andreas Faltz when I was at the MPI, and mentored rates at the MPI, Stan Roberts at Liverpool, and Pat Walsh at Penn, and later, and even ongoing now with Alan Armstrong at Imperial College. And so that's just to talk a little bit about this organocatalysis work that we did with Alan Armstrong and Jordi Burris, who's now at Manchester. He was a postdoc with me at Scripps, but also he had done a short stage in my lab when he was a PhD student in Barcelona. And so, you know, we ended up writing a bunch of articles, and we're invited to write a review to summarize all this work. And it really was based on the idea that, you know, we have these ideas of how selectivity happens in Bronsted acid, co-catalysis, proline provides this acid that helps to guide the electrophile to be on this face, whereas there's a steric model for diaryl prolanol ether catalyst where there's like a blob above the top face, and so you have to attack from the bottom. But some things didn't fit. That's what we called anomalies. There's a few cases that you couldn't explain it by this model. So we came up with we did some experimental work, and we came up with a proposal, and I submitted this one to the NIH in the States. And this is what one proposal reviewer said. This is 2011, before our first paper. We just had some preliminary data. He said, this is really far-fetched. These interpretations border on the incredible. There's a real danger here that the PI will place herself in a mechanistic interpretation hole that she will publicly need to dig herself out of. Okay, well, I didn't get that one funded, but we did it anyway. We worked on it, and it ended up with all these papers, and it turns out that it makes perfect sense. And this is something that people have seen in other reactions. Eric Jacobson's manganese-salem epoxidation has this idea of what Eric calls hierarchical selectivity. You know, the models that we talk about up here, this is the first pass at deciding, the first selection level. And for many reactions, that's all you need is this simple model. But then there are some reactions where at a step later, you can actually bifurcate and make some of the wrong enantiomer, even after you've made the decision up here to make the correct one. So the channel that decides here can be either eroded or accentuated by what happens down here. And it seems like it's an important thing to know as a design parameter, because if you're designing a catalyst, if you're only thinking about the first level, you might miss important features of the second level. And that's something that we worked on as well. basically, there are a number of these interesting organocatalytic reactions are run in cascades. Dieter Enders had this, one of the first papers where he's got 3 different cycles here. You do enamine catalysis, iminium, and then enamine again, and you set 4 stereocenters in one pot, and it cascades. It's like a relay. I think of it, this is his diagram here, and this is my version. I think of it like a relay race. You know, you've only got one catalyst in there, and you've got to keep handing the catalyst off like a baton to the next runner. And so one of the questions that comes up is, you know, how do you make them go in the correct order? I mean, you don't want substrate C to start reacting before A and B. And so we developed, you know, an understanding of what you need to have to make the first step always be the first step. And it really starts to talk about complexity and even emergent behavior, because you've got this system with all these moving parts, and you want it to go, you know, you're giving them marching orders, and how do you make them, you know, stick to their marching orders? So, so that actually brought us back. I mean, just these ideas of complexity brought us back again to origin of life research. And we actually got into, you know, I talked about autocatalysis, which is kinetics, chemical reactions. You know, we didn't have to think about physical phase behavior at all. But it turned out through some of that, some of the organocatalytic reactions, we kind of had to start thinking about phase behavior. And we got into it, you know, you get into an area you don't know much about, you start reading. We were looking at ternary phase diagrams. Martin Klusman, who left my group and was at the MPI in Mülheim for a number of years, came up with, you know, we came up with 2 different models actually that have to do with phase behavior and the way chiral molecules crystallize. And the first one we called crystal engineering, and the second one I named it chiral amnesia. Jack Dunitz told me that's the most forgettable name he ever heard, but I still like it. But anyway, so we published, and these actually work for 2 different forms of the way chiral molecules crystallize, one for each. And there was one of our papers was written up in Nature has a section on news and views where they talk about different papers, and the writers of this, Mike McBride and John Tully from Yale, said this is the first original method for isolating single-handed crystals since Pasteur used tweezers, which we thought was a really nice thing to say about our work. And then there was a Nobel workshop that I was asked to speak about at the Royal Swedish Academy of Sciences on the origin of life. And Leslie Orgel, late Leslie Orgel, he died a couple of years after this workshop, who's, you know, huge in many fields. You know, he started out as an inorganic chemist here in the UK, where you guys are. But anyway, he stood up after my talk and said, well, the chirality problem is solved. You know, we have everything we need to understand the origin of chirality. And so I was talking to my son, I think he was like 11 at the time, and about this, because I was kind of proud that somebody's saying something nice about my work like that. And he said, well, does that mean you're out of a job? If they solve the problem, do they still need you? So anyway, we haven't completely solved the problem. We're still working on it, and it's fascinating work, because it brings in chemistry, you know, organic chemistry. It brings in physical phase behavior, physical and chemical aspects of matter. Obviously, if you think about it, probably, you know, not one of the. You have to think about all those things to solve a problem like how did life start on Earth? Okay, so I think, I just wanted to have one last slide about that, because we've been funded through the amazing foresight and generosity of Jim Simons. Jim Simons he's now just stepped down as the head of the Simons Foundation, but he was a mathematics professor. There's actually a good biography of him. If you Google it, if you're interested, you could read it. He started out as a mathematics professor. He was chair of the department at Stony Brook in New York. He quit and started Renaissance Technologies hedge fund and made $15 billion. Okay, so he then spent the rest of his life funding research, all kinds of different research you can see. And a few years right before we started our work, we were working on this anyway, but he invited a bunch of Nobel laureates to a retreat and asked them, what are the large, fundamental, unanswered, underfunded questions in science? And Jack Szostak from Harvard pitched this idea of origins of life. And they invited 10 people to the Simons Foundation. I was one of the original investigators. And we just sat, it's astronomers, geochemists, RNA world people, prebiotic chemists, very wide range of aspects of origin of life. And we meet there 3 times, before the pandemic, 3 times a year, and he supports our research for all the PIs. So he's somebody that has a mathematical theorem named after him and an asteroid named after him, and he supports basic fundamental research. So I just like to give a shout out to Jim and Marilyn, because they have been incredibly supportive of our work. Okay, so I showed you scripts at the beginning, so I'll just give you pictures of the other places I've been on this ride, Pitt, Merck, MPI, Hull, and Imperial College. And I'll do one more timeline of first my group. That was our last Christmas in person. Last year we had to do it as a Zoom meeting, but we still managed to trade presents over Zoom, did a stealing Santa. And then the last timeline of the other, I already mentioned my son who's been on this journey, and just some pictures of him over time. He's now a musician in Liverpool. And we were at Scotland just right after they finished filming the first Harry Potter movie, and the engineers on the train let him get in and shovel coal on the Hogwarts Express. So that was a nice trip. So, and I'll stop with this last slide, just because you may have seen in my title slide, I'm called the John C. Martin Endowed Chair, a chair that was endowed by John and his partner Lily Liu. This is a man who saved millions of lives with the therapies that he developed for HIV, and his work cured HCV. He was chairman and CEO at Gilead before stepping down, but tragically this end of March this year, he died. And we can't thank him enough for all the support he's given to Scripps and to my lab. So, so with that, I'll stop, and I'll be glad to answer any questions.
Thank you very much, Donna, for such a nice overview of your career over the years. So I'll just wait and see if we have any questions on the Q&A box here, of which yes, go ahead, yes.
I'd like to ask Donna the same question I asked Rebecca, which was that, is there any advice that you would have appreciated at the beginning of your career?
Yeah, and I'm not sure. I guess I would say the most important thing that I found in my career was that whenever you have a big decision, I mean, I had lots of big decisions where I changed, from academia to industry, and then I moved across the ocean. What you have to do is look at all the data, you get all the data in front of you, take everything into consideration, not just the career. I mean, many people will be involved in two-body problems, and you have to give the proper amount of weight to everything in your life. And so don't think, oh, I should take this job because it's really great, but it's not good for my partner. You make those decisions, and then you try to make it work. And most of them in my career ended up working. But if it doesn't work, I didn't mention, I was at University of Essen when I first went to Germany. That didn't work, and I immediately found something else to do as soon as I realized it wasn't working. So make a decision based on all the things that are important to you. Work as hard as you can to make that decision work. And if it's not working, if you give it enough try, if you work hard enough at it, make another decision. And don't be afraid to make big leaps. And don't listen to, listen to everybody, but don't take on board, advice that comes from sources that you may not trust. I mean, when I left academia to go to Merck, everybody's told me it was a big mistake. They told me, your career is over, you're going down a black hole, no one will ever hear from you again. I think that turned out not to be true. And it turned out that experience at Merck was a defining experience for me. I would not be doing the work I am now. I couldn't have learned that much organic chemistry by staying in academia and collaborating. And I couldn't have had that perspective on problems that face the pharmaceutical industry if I hadn't gone there. So I'm a big fan of trying, working in both, actually working in government labs, in industrial labs, and in academia. I ended up back in academia, but where I am now is almost like, we've had 70 startup companies from Scripps. We've got 11 FDA approved drugs were discovered at Scripps. So in some ways I get the best of both worlds, but you've got to figure out what world is important to you, and trying lots of different things is a good way to do that.
Thank you.
I'm just having a look at the Q&A box here. We don't have any questions at the moment, so I don't know if people are still typing the question, but feel free to drop one or two more questions. But on a yeah, and then yeah.
And I don't know if you do, I've been at several of these sort of Zoom meetings where people can type questions and you can send them to me later if they end up typing it in after, you know, or send questions in some form later if things come up. I'm happy to do that.
Oh, okay. I'm sure Josie can arrange that, yes.
Yes, of course.
Yes. Maybe just one. Oh, yeah, go ahead, Josie.
I was just going to ask, because you do have the experience of the industry and the academia that still I don't think is that common today. Is there any key differences that make a big impact in people's sort of work? What people might like?
Well, I think that, so the biggest thing that hit me right away when I got to Merck was, before that I was a professor. You do some work, you go to a meeting like this, you know, you talk about your work at meetings. Well, it used to be in person all the time. And, you know, you give your talk and everybody claps at the end and you sit down. And you go to Merck, that's not your currency anymore. It's like, they can clap, but what I have to do is deliver, you know, a kilo of the API for the clinical trials by the end of the year. You're in research, but you're actually in production if you're in pharma, because you're supporting the clinical trials. So the big thing I learned was teamwork. You have to work, you got to work with people. This is the molecule, you guys got to make it, get together. You can't, you know, I don't like working with that guy. I don't, you know, and you can't, you know, looking and sounding smart doesn't go anywhere. You doing, you know, showing something, you know, basically doing it. And that's where the teamwork, just you've got to use all your resources, and the teams tend to be pretty big. I mean, at Merck, we had a team of chemists and engineers that had to work together. And my group was sort of outside of that. You know, they would come to us with special problems, but you know, learning, being a good team player, learning how to play well on a team really will help you all through life. It'll help you with your children. You get married and have kids. But so that's probably the biggest thing. And then, you know, the kind of leadership skills that you need to run a project like this. I think that's much easier to learn in industry than it is in academia. It's just you have to do it kind of by default in academia, but you know, you can get away with doing a bad job at leadership or teamwork in academia, and you can't in industry.
Thank you.
We have another question from Nakisha Mark, who's saying they really enjoyed your talk. So thank you very much for that. And they're saying they made reference to your work in their thesis, which I'm sure you are used to. So the question is,
that's very nice. Thank you. I appreciate that. So yeah, my son was born. The question is about making career moves and having a family. So my son was born when I actually, it's a long story, but I went to Merck when I was 5 months pregnant. I left my academic position and started at Merck. And so that had its own whole set of, like, people thought, who is this? We don't need this fancy chemical engineer coming from academia, and she's just going to go on maternity leave anyway. So that was a bit of a challenge to be taken seriously. But he went, you know, Merck had this fantastic daycare center. It was right on site. I could go and see him during the day. So he was there for 2.5 years. We moved to Germany when he was 2.5 years. And then, you know, we plunked him down in this other environment, and, you know, he was just starting to learn to talk well in English, and all of a sudden everybody's like, so for 3 months he didn't say a word, and then he just was speaking perfect German. And then he would tell me, when I take him to the kindergarten, don't talk to anybody, Mom, you know, because my accent was so bad. So I understand how immigrants feel when the parents sort of, like, always, you know, the kids are sort of ashamed of the parents. But you know, I think it has to be you all have to be in it, you know, together, and it's an adventure. And I think it's a fantastic experience for kids to see parents who are happy with what they do, and that they have a purpose in life. I think that if you can show all those things, even if you don't move around the world, you can show your kids that they're very important, but also you're as a professional, that you have important career too. So yeah, the tips are that you should try to be, you know, on the same page about how you do all these things. and never, I mean, you shouldn't like apologize and say, oh, you know, I wish I could stay home with you. You say, look, you know, mom has her own stuff she's got to do. And just like you want to go play with your friends, you know, she's going off to play with her friends during the day. And if they think of it like that you're not feeling guilty. I mean, if you transmit the feeling of guilt about being away from your child, you know, they will pick up on that. And so, you know, try to work out the best balance between work and not work. Maybe for some people it might be part-time, works out better. Or, you know, make sure if you're in a relationship that both partners share in all the work, that helps a lot. But I think just being honest, you know, with your kids and involving them in decisions if they're old enough is a big first step. And don't think you can't do both. You know, you don't have to be, you know, I'm not talking about superwoman having it all, whatever, but you can have a career and a family. And there will be some sacrifices that you have to decide which ones, but don't let anybody tell you that you can't do both.
Thank you very much for that. Maybe just one last quick question. It's interesting to see how you don't really, how the reviewer comments don't really bother you that much. Not really bother, but you take them. I mean, I remember a few of the comments here on the applications you made and how the reviewers were really not into it, but it didn't seem to bother you. Is it something you've learned over the years, or you're just confident on the work that you do?
So I'm not sure, I couldn't hear very well everything you said. It was breaking up a bit, but I think you were asking about the, like when I was talking about the proposals and what the reviewers said.
Yes, the grant applications, yes.
Yeah, so
I remember quite a few of them, the comments were not so good, but
and that's
bother you.
But I really wanted to put that in there. So, because when you see someone give a talk and you see all this work, it's done, it looks great, you know, and you never see all the stuff that went on behind it. And everybody has, so it looks as if no one has those problems, right? When you're showing a nice talk with all these great results, but almost everybody has these experiences. And so it's just good that everybody knows it. So it doesn't sound like, oh, she never has any issues. I mean, I tried 6 times in a row for an NIH grant in the US. You know, and so, you know, you have to be creative about, and you have to have a thick skin, you know, because reviewers, especially when they're anonymous, you know, they can sometimes, they'd say stuff they would never say in person, right? But don't take it to heart, especially, you know, I mean, sometimes they have constructive things to say that you can make a better proposal or a better paper, you know, and that happens almost all the time. There are constructive things in these comments. It would be nice if they didn't say it in such a mean way, but everybody, this happens to everybody. So don't feel bad is the first thing. And then look through it. Usually it takes me a day or two. I get it. It's like, oh God, and I put it away. It's like, I can't look at this. And then I come back a day later and then I try to look through what was reasonable about what they said. And will that make usually for papers, you know, will that make my paper better? Or, you know, with proposals, how can I write this in a way that, you know, one of my issues has always been, how do I write to get organic chemists to pick up on what I'm saying? Because I'm an engineer and I don't have that training. Alan Armstrong at Imperial has been teaching me, helping me a lot. But, you know, you have to craft your paper or your proposal in a way that the people that you need to see it are going to read it. So, you know, write it, put it away, come back a day later and try to read it from scratch to make sure that you're saying, you know, what you need to say in the order that you need to say it in and make it, you know, as clear as possible. I think that's, you know, and don't be afraid to edit. You know, it's actually what writers tell aspiring writers. Doris Lessing used to get people sending their manuscripts and asking her for help. And she'd say, the one thing they never wanted to do was edit and rewrite. And that's the one thing you have to do. Like if you're really wedded to an idea and somebody shows that it won't work the way you want, you got to take that in and you got to, you know, re-gauge, you know, your, you know, what you're going to do. So you got to be able to take criticism and to let some of the stuff just roll off, you know, the mean stuff roll off. I mean, everybody goes through it. So don't ever think you're the only one. I mean, that's what I used to do. I just want to crawl into bed and pull the covers over my head when I get a bad review. But it's better, you know, that's why I decided to start putting these comments out, you know, in a talk so everybody can see, you know, because the day you get that report back is, you know, is a bad day. So you can make something good out of it in the end.
Oh, okay, I think that's it. Thank you very much, Donna. There's no more additional questions.
Thank you very much for the invitation.
Yes.
Thank you.
And thank you for all today's speakers for a really excellent programme. It's been fabulous, very thought-provoking and very inspiring. So thanks to all the speakers today. We hope our next event of this kind can be in person, and then we can invite all our fantastic speakers to come and stand in front of the lecture theatre and talk to us. But we can only control what we do, so we just have to hope on that one. But yes, hope to see you all in person soon.
Thank you.